Okay, our next presenter is Stephen Blaine. Stephen comes to us from York, Maine. He has a Bachelor of Science in Wildlife Ecology from the University of Maine. Like Gabriel, Stephen has had a range of field jobs before coming to Archbold. He joined us here in May 2021. Yeah, and his jobs range from having done research on amphibians for the USGS, working at doing small mammal and carnivore work for the University of Maine, post baccalaureate, and he's widely traveled. One of the fun things about working with Stephen is getting his weekly reports on where he's traveled over the weekend and what interesting new natural history he's witnessed. Stephen is going to talk today to us about some new novel methods that he's developed here at Archbold for understanding small mammal trapping and small mammal presence here at the station. So without further ado, I'll turn it over to you, Stephen.
Cool. So my project was sort of an evaluation of implementation of camera traps for detection of small mammal communities. And when I started out, I wanted to look at how the different burn unit management fire regimes affect the small mammal communities. But I didn't really end up getting to explore that too much, and you'll see sort of what I ended up doing instead. So the first thing is why I would want to look at small mammals when we have so much data about sort of medium and large mammals from the ranch. And I guess a lot of it has to do with personal interest and a background working with them. But I also think they're really valuable ecological indicators in ways that sort of larger mammals, I shouldn't say aren't, but they sort of have different value in different systems. Small mammals, of course, are really good connectors in terrestrial ecosystems, and they act as sort of, obviously prey for predators, which I was able to get some photos of, but also, they're seed predators themselves, they're insectivores, they sort of affect the soil structure. They just have a lot of really important functions in ecosystems. And because of their small home ranges and really high reproductive potential, they sort of can inform you about things happening within a system at a really small spatial and temporal scale, in ways that, like a large carnivore sort of would have more of a lag time if you were studying that population. So I guess an additional value to monitoring this extra community within an ecosystem. So to do this, I had to modify camera traps quite a bit. This is something that's been done a number of times now and seems to have started actually in Florida. There are a couple of early papers from, I think, like the early 2000s of people using buckets in camera traps to look for small mammals. There's designs that float, there's designs that go under snow now, there's all sorts of modifications. And I ended up using basically what these two papers did, which is a 5-gallon bucket with a camera attached to the lid pointing down, and holes cut in the sides of the bucket so that animals can pass through. So it's a trap just like a camera trap is a trap, not in that it catches anything other than on photo. So nothing gets stuck in the trap. Things can easily come and go as they please. But basically having the camera contained allows me to set the focal distance, and it keeps bigger animals out because I did put bait in some of the traps, and it just helps make the photos easier to see. So my sampling scheme was I sort of set out to get different units with long and short fire return intervals. And I did that sort of in a stratified sampling scheme of different habitats, sort of coarsely defined on the station. Because I only had like a certain number of cameras out at any one time, I ended up using like different months as replication, even though there's, there are different times a year. I figured that the variation, especially if just looking for sort of the presence of a species, wouldn't be too much. Just sort of, an assumption, but I think it worked out. The one thing is, if I was going to consider abundance, I think that would have to be something I'd have to be more careful about, because there is like a seasonal increase and decrease of a lot of small mammal abundances. I had 18 cameras, and at any one sampling period, they were split up into 6 sites with 3 cameras at each site. 2 of those cameras were at either end of a drift fence, and one was sort of a short distance away with bait in it. And I use that 3-camera unit as sort of a sampling site. So all of my data is pooled by sampling site. It's not unique to different cameras. I put the cameras out for 3 weeks, and I tried to time it roughly with lunar cycles. So it was like 3 weeks starting about a week after a full moon. And then after that time period, I'd take them in and put them out at a different site. This is a map that sort of has my clusters of sites and the units they were in. The 2 sites to the north are, well, the one farthest to the north on Red Hill is sand pine scrub. The one to the west of that is scrubby flatwoods. And then the site to the far south is all seasonal wetlands. And the site above that was oak hickory scrub. In the end, I ended up only analyzing a small set of my data. Basically, I took half of the array sites and left out the baited camera traps, which had about twice as many photos as the non-baited ones, and just analyzed those photos. So it was a total of 255 trap nights worth of images from 12 stations, totaling 36 cameras, and I detected 20 species. This is just, a sample of what I saw from those cameras. I ended up actually getting more species than that in total, but many of them are one detection for the whole 4 months that I had cameras out. So in terms of analysis, it doesn't really amount to much, but it was cool to see how many things sort of got picked up, and a lot of things that didn't. I sort of expected, like towhees, there's a lot of common things that didn't show up that I would have expected, and then some really uncommon things like the long-tailed weasel there in the bottom row, the shrew in the top, that I sort of was hopeful to see but didn't really expect to. So yeah, it's pretty interesting. These are sort of an evaluation of how effective I was at detecting. Well, effective I was at designing the detection for the species, because I wasn't sure how long I needed to leave cameras out for small mammals. There's a lot of literature about how long to set cameras up for like large carnivores and stuff, that it's more of a standard method to search for. And these graphs just show on the x-axis, it is the days that the cameras are out. It's not the trap nights, so it's just for the unit. It's not for each camera, but the days the camera are out, and then the number of species that the cameras detect on the y-axis. And it's sort of, as the curve approaches an asymptote across, that's sort of the amount, it's a way to evaluate the amount of time you're going to leave out cameras for detection of species. And obviously tends to be that the species further to the right on the graphs are sort of the either less common or less detectable species. So depending on what you're looking for, it sort of gives you an idea of how long you have to have cameras set up to be effective in your surveys. And I think, it ended up working out that for mammals, I think it was a good time period, but I also got a lot of reptiles, and it's a bit difficult to tell because the detection on the cameras is based on heat a little bit. So I got a very small representation of the snake community here, but I did get a lot of the big diurnal snakes that would be warmer when they pass the camera. So I'm not really sure if that's a product of not detecting them as a product of me not leaving cameras long enough for them, or if it's to do with them not being picked up by the cameras at all. or of course, any other number of things that might have happened. I ended up not analyzing enough points to really get a lot of statistically significant data. But I did sort of find trends that I'd like to investigate further with more data and sort of follow what I was able to intuit just by looking through the thousands of pictures that I got. So this is golden mice occupancy and detection probabilities by habitat at the different sites that I analyzed. And as you can see, the occupancy is sort of all in one habitat, which isn't quite what happened on the cameras, but it's pretty good. I think there was 1 or 2 sites in Oak Hickory Scrub that had them. But in terms of the number of detections, it pretty well followed that sites that were sandpine scrub had them, and sites that weren't had very few of them or none of them at all. And then you can just see that for all of the habitats except sandpine scrub, where they were fairly common, I have so few numbers of them in the photos that I analyzed that I basically can't estimate the detection probability at all, which again shows you how few there were in those habitats, which implies that they're not very common there, as I was able to detect them with the same methods in sandpine scrub. Similarly for spotted skunks, the significance of the occupancy and detection probabilities is basically non-existent with the sets that I actually was able to analyze. But I did find, as is true in all of the literature, that they have very high occupancy. They're just extremely difficult to detect. They have a really low detection probability, and it takes a long time to get them on cameras usually, or in traps. And so I was sort of surprised to find that, because I didn't really, it was unclear to me how common they were here. Though I had seen that Dr. Lane did catch them sort of regularly, he didn't catch many of them. And so I was interested to see them sort of in a lot of different sites, but usually only one detection. And then finally, I looked at old field mice because I actually did get sort of enough numbers with them in the subset that I analyzed to get kind of results that are statistically useful. Oops, sorry. The occupancy is sort of the same across all sites. You know, they weren't in the seasonal wetlands, which isn't a big surprise, and they tend to be everywhere else. But then detection probabilities varied quite a lot. And as you can see in the Oak Hickory Scrub, the detection probability is actually statistically less than it is in the sandpine scrub and the scrubby flatwoods. And to be honest, I'm not sure what that reflects in my methods, because the camera traps, sort of the immediate area covered by the camera trap, should have equal detection, obviously, you know, which is not the detection probability. But it may have to do with food. It may have to do with sort of a more complex habitat, because those sites had a lot more dense sort of woody debris and stuff. That maybe the drift fences were less of a direction for small mice. But anyway, I would need to do a lot more investigation to get much more out of it. But and then basically at the point I'm at now, a lot of what I got out of my study is sort of more questions, which I feel like is normal. But there were a couple things that came up as a result of collecting data that I'd really like to look into further. I think there's a lot of potential for using the non-invasive camera trapping method with small mammals, coupled with sort of a less intense than normal live capture. I think it would allow you to mark individuals. It would allow you to sort of get, you know, breeding status and body condition from individuals and get sort of baselines that you could then sort of figure out a way to quantify with some sort of like ImageJ perhaps, or there's a lot of software that you can use to take images and sort of get metrics from those images once you have an index to base on. So you'd have to periodically do live trapping, but you wouldn't have to live trap, you know, several times a year to get this information. Also, I got a number of animals that were individually marked naturally, like this rice rat on the left, with an injured eye and a broken tail. And it actually, I got it on multiple sites, which I was hoping to avoid with my sampling scheme, but having the marked individuals showed me sort of the movement of the animal in habitat. So that's potential for this. You could set up cafeteria experiments where you have different food choices in the base of the bucket and sort of monitor what different species go for, which would inform, perhaps like possibly plant community structure as shaped by seed predator rodents. There's a bunch of interactions, both intra and inter-specific with the rodents, especially around bait, which is not surprising. There's just I think there's a lot of potential to couple this method with sort of the more traditional invasive method, and get a lot of new information out of it. And that's it. And I don't have a thank you slide because I was working up to the line here, but I'd like to thank Joe and Raul from Predator Prey Lab and the USDA Feral Swine Project at Buck Island Ranch. I'd like to thank Gabriel and Sharam, and everyone else at Archbold and at the ranch. And yeah, it's been great. And any questions?
Thanks, Stephen. We have a question in the chat here. I'll throw at you. Joe Gentili asked, did you reuse any of the sites from Jim Lane's past small mammal grids at Archbold? If you, I'll go ahead and I'll move to the next question after you answer that one.
Sure. I spent a lot of time sort of trying to plan that out, and I would like to have done that. I didn't use any actual specific sites. I did get, I think I was in like an adjacent burn unit for one of my sampling periods, and it's, according to the habitat maps, it's the same, broad category of habitat.
Mm-hmm.
But no, I didn't use any specific sites, and because of the small mammal sort of highly localized, especially without bait, use of habitat, I am not sure that I can really say anything about it. I also wanted to just get into his data more, but I haven't even gone into mine as much as I would have liked. So, but I do think there's a lot of potential for interesting comparison against his, especially for, I guess, some of the less common species, because I don't really have any sort of abundance information that I can draw from mine. But I think he obviously did because he was live trapping. But it would be interesting to see sort of the different community structure that I'm getting as compared to what he got. And there's a whole bunch of fire history in between there. It's sort of an endless, like the pigs on the ranch, an endless data set with lots of interesting questions.
Just qualitatively, can you say that based on your knowledge of what's in the data, are there any characters that are kind of obviously missing, or perhaps more present in the record now?
Sure. So this is not an analysis I've done. I was surprised at how many old field mice I got, as compared to basically in the stuff I was seeing from Dr. Lane's live trapping, which I think I was looking mostly in the 90s. It was a really high percentage of Florida mouse and cottonmouth. And I sort of got more, they were still the dominant taxa in most places, not in wetlands, not in every single trap station on some of the other habitats, but usually they were. But I got sort of a lot more old field mice than they were recording. I got a lot more rice rats than they were recording. But that might be placement, because I don't think that they were going in wetlands like I did for that one sampling period. Fewer cotton rats. It seems like I got a different mix kind of across the board. I got, I'd say, more skunks and more weasels, but as a percentage, but my traps have a pretty big hole, and I don't think the small mammal live traps, it's a tight squeeze for the bigger stuff. So
how many species total did you catch on the cameras? Is another question we have in the chat.
So in the ones that I analyzed, I got 20. I can think of like 7 or 8 more sort of immediately. And there are a few like bobcat and armadillo that I only got once. So there's, yeah, I don't know. There's potential for some that I don't even realize that I got. But yeah, I would say probably like between 30 and 40.
Gotcha.
I could find that actual number if anyone would like it.
Were there, Raul asked this question in the chat as well, were there any species that you would have expected to find that you're not at this time aware of?
So like I mentioned the towhee earlier. There are some like so in terms of small mammals, I would say that the only, there are supposed to be a few shrews here, and I think I only got short-tailed. I wouldn't necessarily expect to see the other ones. I was sort of happy to see the one, but I think that's of note. I don't know when the last time someone saw like a southeastern or a least shrew here. In terms of herps, I sort of thought I would get more snakes than I did. I only got 3 species, and one of them was a single pine snake. So it was all racers and coach whips. Birds, I thought I would get more of the, like the towhee. I thought I would get maybe a blue jay or a scrub jay, or because I did use bait again, or like any woodpeckers. But in terms of mammals, I think I got pretty good coverage of what I expected. I did sort of, I was hoping one of the wetlands would turn up a round-tailed muskrat, but I never got any.
Gotcha. Betsy Roswell asks, which habitat, and maybe you can speak to this, which habitat had the highest species richness for mammals specifically? Can you say that based on what you have?
Yeah, so it was the sandpine scrub. Yeah, it's also true.
Not the oak hickory scrub.
Right. Yeah, because I was attempting to sample the different burn regimes, the sites for oak hickory scrub, one of them, I think the long fire return interval one was a good oak hickory scrub. There were lots of big, good sized trees with lots of mast. But the short fire return interval sites, I think the fire, even though I think it was 3 years ago, it might have been 4. I think it was a pretty intense fire, and there were, the trees were all sort of shorter than me. So that might have affected the overall richness, just because I had fewer cameras out in sort of sites characteristic of that habitat.
Yep. Got you. Well, I have a couple of more here if you're still game.
Yeah.
Maya asks, why did you set the traps for the week following full moons?
So there's a lot in literature about small mammals being less active around a full moon. And the reason I did it a week after instead of 3 or 4 days after is because I was not ready at that time. So a little bit of just convenience, and also I was trying to avoid the full moon basically, and cycle based on the the lunar cycle.
I see. Then here's another good question about kind of camera operation from Amartya. Is there any chance of mammals being camera shy, where they might avoid white flash cameras?
Yeah, yeah, definitely. I was sort of worried that the confined space of the bucket with the flash would be a problem. But after the first sampling period, I mean, there are pictures of rodents, especially, I guess, sort of standing at the entrance. But they usually end up going in anyway. So I guess the flash is not enough of a deterrent to keep them out of. I mean, certainly with bait, but even without the bait, they seem to be willing to go in. And I think just the fact that there's no, I think the flash occasionally startles them, but they'll still, most of them at least, will still go in because there are very few pictures where they stop at the entrance and then never go in. Yeah.
Got you. Well, that brings us to 4:30. If there are, here's one more from Raul. Asks, what did you do to diminish the flash brightness? Wonder if you could talk a little bit about that.
Right. So I guess I sort of skipped over. To put the cameras in the buckets, I modified them in a couple different ways. Because I was worried about the flash, I did cover the flash with paper. I think I started with like 4 layers of printer paper to sort of diminish the brightness, which both for the animals and so that the pictures weren't just completely overexposed every time. Because it's, instead of, the flashes are designed for probably like 10 feet away from the camera sort of thing. And in the buckets, they're less than 2 feet away. So
yeah.
And I also put close focus filters over the lenses so that the animals on the surface of the bottom of the bucket would be in focus. Again, the cameras are sort of designed for, I'm not actually sure what the optimal focal distance is, but it's probably between I'll say 5 and 15 because that's pretty safe.
Yeah.
Away from the camera. So to bring the focus a lot closer, I had to put close focus lens over the camera lens.
Got you. Do you think that the trap that you've designed, kind of developed here, can be used, is useful? I'm sure it can be modified in other ways, but did you kind of like the way that it worked?
Yeah, I did. There's a couple, I would make some changes, but I like it as a general sort of surveying technique. I think so. The pictures on this final slide, the one on the right of the Florida mice, young and older, it's actually, when I was planning the designs, I wanted to do a design with pictures from the side, lateral view, instead of dorsal view, because for a lot of rodents, it's like an easier way to ID them. But I ended up using the dorsal view because there were buckets that someone was kind enough to let me use. So I didn't have to buy anything to set up sort of a different view. But one of my baited traps got pulled over by a raccoon, which is what those pictures are from, with the lateral view. And yeah, they're probably the best pictures I got. So that raccoon deserves some credit as well.
Agents of chaos. Okay, well, we're now a little bit over time. I want to thank you, Stephen, and Gabriel, for your presentations and for your hard work here. It's well done.
Thanks.
Laura, any final notes?
Yes, thank you everybody for joining us today. We had a really good crowd. And join us again next Thursday for a distinguished speaker seminar presented by Dr. Royson Stanbrook. This will be on Zoom again. Unfortunately, I was really excited to do it in person this month, but coming soon, we will have our in-person seminars, but next week we'll be again on Zoom, and I hope to see all of you here. Thank you, and you might see in your Q&A, I'll just leave it on for a few more minutes because Gabriel, AKA Stephen, is typing his answers to your other Q&A questions. So I'll just leave this running for a few more minutes. Thanks for coming, everybody.